Dr. Jeffrey Sundstrom, who's going to be talking about proteomic profiling of ocular fluids and some very interesting work that he's going to talk about. Thank you very much, Jeff.
So thank you, Josh and Benny, for inviting me to speak today. It's quite a pleasure and honor. Before I start, I just have one financial disclosure. I'm the chief scientist for a company that we formed in December of 2024, Ocular DX. I'll be talking about a vitreous biopsy device at the end of the talk, but all the data that I'm talking about has nothing to do with Ocular DX. It was derived from samples from a biorepository at the University of Michigan. So before we start talking about precision medicine and how I think we might be able to get there, I just wanted to take a step back and think about how we got to the current treatment mechanisms in AMD and diabetes and vein occlusions. And so after Judah Folkman identified factor X as VEGF to help cancer grow, Lloyd Aiello in a seminal study in 1994 showed that in ocular fluid, particularly active PDR or patients with iris neovascularization, have elevated VEGF levels in vitreous. I'd also like to call your attention to the widespread and variability within that graph. So yes, on average, there's more VEGF in those patients, but the mean is almost as large. The standard deviation is nearly as large as the mean, and I'll come back to that shortly. So fast forward to 2006, and the FDA approved Lucentis. Eylea was shortly thereafter. And that really revolutionized how we take care of these patients. This was the first time that we could actually make these patients see better. So when they respond well, they can pick up 10 or 15 letters of vision if we can get their macula dry. We do 14 million injections a year, which is just an incredible number of treatments. That is not me at the top right. I wear gloves when I inject patients. However, on the bottom right, that is a patient of mine who had wet macular degeneration. After a single injection, went from hand motion vision to 20/60. That's when it works really well. However, if you look at the Cochrane Review database meta-analysis, on the whole, for these retinal vascular diseases, there's about a 50% response rate in terms of patients that respond really well. So we have a lot of headway to go. And it's always bothered me that unlike every other field of medicine, when we're using a very specific treatment, we're not measuring that target beforehand. Frankly, I'm rather shocked that the insurance company doesn't force us to do that. We wouldn't do this in cancer. We wouldn't give an anti-HER2 new antibody if you didn't express the HER2 new receptor. Couple that with new and emerging therapies. So faricimab was approved in 2022. It targets VEGF as well as Ang2. And in phase 3 trials now as a Wnt agonist, and hopefully another drug targeting interleukin-6 will be coming to the market. So I think we were comfortable before when all we had was anti-VEGF. That is all we would give, and we would just hope for the best. But it's time to move on from empiric therapy and I think move towards a more precision medicine approach. And so how can we really, optimize the right treatment for the right patient at the right time? That's the whole concept of precision medicine. And I think, as we all know, we all have friends and family members, unfortunately, that have had suffer from cancer. The first step is always some sort of a biopsy, be it a tissue biopsy or a liquid biopsy, followed by some molecular profile, and then hopefully some very targeted treatment with better cure rates and less side effects. We don't do that in ophthalmology. The other reason you'd want to be able to stratify patients is you could potentially bring more drugs to market. So the top drug on the right is lampalizumab. They missed their mark. I think their p-value is 0.06. I know the former CEO of the diagnostic portion of that company. They were looking for plasma biomarkers, not biomarkers in vitreous. Ophtotech was a drug targeting PDGF, and it also missed its endpoint by very small margins. It's been shown in the literature, your odds of making it to FDA approval go up at least twofold if you have a biomarker. More recent studies suggest that number might be closer to 4 to 6-fold. So how can we move towards precision medicine and ophthalmology? I'll briefly want to thank the University of Michigan Kellogg Eye Center. I started a biorepository there as a fellow. We have about 3,000 or 3,500 samples there, and those are the samples that were used to create the data here. We're creating a similar biorepository here at Shea. So I'm going to talk to you about some vitreous proteomics work that we've done and then introduce you to the vitreous biopsy device. So obviously, unlike cancer, we can't access my primary tissue of interest, which is the retina. We can't do a retinal biopsy. So we need to look at proximal biofluids, or as Eric Pearson mentioned earlier today, I think a few other people, liquid biopsies. And so the project that we started more than 10 years ago was trying to validate vitreous as a proximal biofluid, and then determine if it is a proximal biofluid, the profile, the molecular profile should change between disease states. So this was one of the earliest studies we did. Back then, it was about $5,000 to run one sample on a mass spec instrument. We took 5 patients, pulled them together, proliferative diabetic retinopathy and control, and found over 1,000 proteins. And then we did an unguided pathway analysis. And sure enough, it told us that there's proliferation of vascular cells and neurodegenerations, the 2 hallmark aspects of diabetic retinopathy. In addition, it suggested that a new protein, Wnt, might be involved. I shared this data with my former PhD advisor, David Antonetti, who went on to do some outstanding preclinical work showing that the Wnt pathway is involved in animal models of diabetic retinopathy. Tony Adamus and David Geyer started a company called iBio. And after a phase 1 trial, this was purchased by Merck last year, and it's now in phase 3 trials. We've since gotten much more sophisticated, but we can do 10 samples at a time. We've driven the cost down to $500 a sample, so now we can do on an individual basis. And we've actually done a power analysis, which is critical. So to do discovery proteomics with mass spec, you only need about 20 to 30 patients per group, assuming your effect size is on the order of diabetic retinopathy. But we wanted to get a little bit deeper. We weren't quite rich enough with our pathway analysis, and so we transitioned to an Olink's proteomics platform. The beauty here is you only need 2 microliters of fluid, and you can identify up to 5,000 proteins. So we optimize it for our vitreous. There's some very vigorous ways, which I won't get into today. But applying the same samples that we ran on mass spec to Olink, we identified another 2,300 proteins. Now when we run our eye pathway analysis, as you can see on the right, the prediction is that the upstream regulator for this change in differential expression in protein is VEGF. And there's over 60 proteins that are differentially expressed downstream of VEGF in these samples. And the same could be said of interleukin-6. The point I'm trying to make is in terms of Wnt and VEGF and interleukin-6, we can potentially find novel targets in ocular fluid. As I've given these talks and people have become more interested over time, people have started looking at aqueous because it's easier to get aqueous. You can't just stick a needle and get vitreous in a clinic. There'd be a high risk of a retinal detachment. This is a study that we did recently with post-mortem eyes, paired aqueous and vitreous from the same eye, 16 patients. And these were all obtained within 4 hours of the time of death. And so you can really look at this as a model of ischemia, if you want. And so what's interesting to me is of all the differentially expressed proteins, 1,000 of them were more abundant in vitreous, and about 100 were more abundant in aqueous. So aqueous might be a valuable fluid. Vitreous might be a valuable fluid. It really depends on your question. If you look on the right-hand side, sorry, I should have made the letters a little bit larger, but the retinal ganglion cells are at the top and RPE are at the bottom. So both fluids contain, in at least half of the samples, proteins originating from any given cell type. Certainly, there's more proteins coming from vitreous, or they're more abundant in vitreous than aqueous, but you can see them from any cell type. And then when you do the cluster analysis that you can see here, on the left-hand side are the proteins that are more abundant in vitreous. And we've done using single-cell RNA sequencing, we're trying to predict what cells they're coming from. And the most obvious possibility was that these were cells from the posterior polar retina. And that's what we're seeing on the left-hand side. Sorry, my diagram got confused. On the top right are the proteins that are more abundant in aqueous, and they're actually coming from cells in the front of the eye, so the cornea. The left-hand side is more abundant in the vitreous. They're coming from retinal cells. So all of that being said, we can now identify over 3,000 proteins in a fluid that I was told was protein poor, protein deficient. It just had collagen and glycosaminoglycans. We can find disease-associated proteins. And I think if we had access to vitreous that was safe and cost-effective in a clinical setting, I think we could move towards precision medicine and ophthalmology. Just briefly, I'd like to take a step back and ask the question, why is vitreous so information-rich? And Eric Pierce touched on this earlier today. Went back to our earlier data. We did gene ontology analysis. There was no molecular function or biological process, but 2/3 of all of the proteins in every study I've ever done are EV or exosome associated. And as Eric mentioned earlier today, EVs are critical mediators of cell-cell communication. And so when you're looking at vitreous, even if you're doing proteomics on total vitreous, you're listening on the phone call between different cells that's going on between the different cell types in the retina. So what we found using NanoSight is that there's over 5 to 10 billion EVs in each of our eyes. This hadn't been shown previously. I'm not sure if I'm going to be able to get this movie to play. That's okay. And this is a diagram showing EVs as critical mediators of cell-cell communication. I won't get into that more because that's already been talked about quite a bit today. So studies are ongoing, and we've had some very promising results recently. Just in the last couple of weeks, we're able to isolate EVs from very small amounts of vitreous, probably 50 microliters. And so that'll allow us to do EV proteomics and transcriptomics on individual patient samples. So the last part of my talk will be to introduce a vitreous biopsy device. When I was a fellow, we conceived of this idea. There was an instrument called the Intractor, and it never took off. It was supposed to be a tool to get vitreous sampling done in the clinic, but you had to buy a $100,000 piece of capital equipment. There was all this other material and it never worked. And so we actually had some really basic design requirements, and it had to be office-based, no additional capital equipment, an extremely simple workflow that fits in. Physicians don't like to change what they're doing, especially retina private practice. They're seeing 60, 80 patients a day. We're not going to change their behavior. This needs to fit into their workflow, and it does. It obviously needs to be safe. That's first and foremost. We need to get about 300 microliters. We've shown that's safe to collect in the operating room. Obviously, it needs to be reliable. We don't want 50% of the patients not to be able to get fluid out. And then what's nice is the syringe it's on a Luer lock. It can be disconnected, and you can put a cap on and transport it to the lab. So this is me. This is just a mock procedure with a very old model that was just 3D printed. And so this is the exact procedure that every patient goes through for those 14 million injections that they're getting every year. They get a speculum placed in their eye. They would normally get a 30-gauge needle. In this case, it's going to be a 24-gauge needle for the biopsy. You would actuate the device. There's a red light that initially turns on. There's a Hall effect sensor. When it's rotating at 4,000 RPM, it turns green. And then you pull back on the syringe and aspirate your sample, turn the instrument off, and come out of the eye. This is what the device looks like. It's based off of the engineering and mechanics that are happening in the operating room. So there's a circuit board, there's an electromagnetic motor and a battery, and basically there's an outer needle that's fixed. It's 24 gauge. It's got a port in the side. There's an inner needle that's 28 gauge that also has a port in its side. And so as that needle's rotating and those two ports are aligned, if you're pulling vacuum, then you're going to be able to aspirate vitreous. And so I'm going to see if I can't get this one to play. So you can see there, not much vitreous is going in, but as soon as we pull back on the vacuum, you can see we're getting really nice vitreous aspiration into the port of the device. So this is an endoscopic view of a human eye, showing that the device is working. Of course not. So in summary, we believe the vitreous biopsy device would allow sampling of vitreous safely in a clinical setting and set the stage for acquiring vitreous fluid so we can start to identify novel targets for treatment, particularly in early stage disease, which is what every field of medicine wants to do. Getting samples in the OR from proliferative diabetic retinopathy is nice. It helps us understand the disease to some extent, but we really want to treat patients early before a lot of retinal damage has gone on. We personally think that ocular fluid could lead to the identification of some novel therapeutic compounds and targets. And then once we're done with this discovery work, we would certainly need to develop some more robust assays potentially point-of-care assays, maybe lateral flow assays that you can measure VEGF and Ang2 or IL-6 in the clinician's office, much like you do with other rapid tests that are out there now. And these are the project teams that obviously has taken a large amount of people, and this is the lab, some of the folks from back at Penn State, and then moved here in January. I'm thrilled to have Greg with me as well as Harini here today. So thank you so much.
Anybody have any questions for Jeff? Yes?
This seems to be a marriage between injecting the eye with anti-VEGF, which we used to do in the office, and your vitrectomy device with a rotation like a type of phaco. And it's very ingenious. How long has it been around?
We've been trying, we thought about building it for 10 years. We went to Home Depot, bought some pipes and some things and played around. Somebody told me that a 3D printer existed. We printed some peculiar gadgets up. We went through 3 engineers, and somebody by the name of Lauro Ojeda told us he could make an electromagnetic motor for pennies on a dollar. So the other nice thing is that this device would be sold as a single-use disposable device in a kit. It'll be obviously sterile packed. It'll just be sitting on the shelf, so it works right into the clinician's workflow. Yeah, that's correct. And the cost of goods is $50 to $100 to make. And so 24 gauge outer, correct. No trocar needed. We actually had to work with some needle engineers to get the needle to cut and be self-sealing. So, thank you.
I would also say that if you take advantage of our lovely machine shop, you could probably get a better version and get IP on that for pen. That would be fabulous, right? Then we can solve several problems at one point, right? Okay, do you have a question?
I was just wondering, you said that you were working on or using this to also work on therapeutics potentially.
Mm-hmm.
Do you think that this kind of device, in addition to like drawing fluid, could also like inject fluid if there is kind of therapeutic?
So we've thought about that, but those 14 million injections are largely without complications. So we want to keep the cost down. When I mentioned therapeutics, I meant more from a identifying novel targets that are clinically relevant. And so, I was trained in the diabetic retinopathy lab, and the animal models just don't recapitulate the human disease well at all. So anti-VEGF was approved by the FDA in 2006. That's before the iPhone was approved and put on the market in 2007. And so pharma is really looking for new targets for treatment because we don't have good models of disease and we don't have access to tissue that represents the disease from patients.